Hello, everyone, and welcome to our webinar today. We're excited to share some exciting clinical trial updates with you and introduce you to NMD Pharma and their investigational drug for SMA. Next slide. So this is a quick overview of what to expect from today's webinar. We're going to give you a quick review of the FDA-approved drugs for SMA and briefly talk about how they work. And then we're going to talk about where we want to go, which is optimization of the current drugs and then new drugs to meet unmet needs, and specifically talk about drugs that can be used in combination with already existing drugs to achieve even greater therapeutic effect. And then I will introduce you to NMD Pharma and let them share more about their program with you. Next slide. So as you all are aware, there's been a rapidly evolving landscape in SMA over the last several years. We now have 3 FDA-approved therapies that have really dramatically changed the natural history of SMA. We know that early identification and treatment dramatically alters outcomes for people with SMA, and this is most striking when infants are treated pre-symptomatically. But we also know that treatment of symptomatic individuals can provide improvements, including increased function, stabilization, or even slowing loss of function. But we know there's still significant unmet needs for all people living with SMA, and especially for individuals for whom treatment wasn't available prior to their symptom onset. And we also know that 30% roughly of all SMA patients remain untreated. Next slide. And so this slide represents our current SMA drug pipeline. You can see in the box there are the 3 FDA-approved treatments, Spinraza, Zolgensma, and Evrysdi. And then you can see the variety, the breadth of drugs that are in various stages of clinical development, so different phases of clinical trials, and then those that are in preclinical discovery or different stages of development to get them ready to transition into the clinic. Next slide. So I want to step back for a minute and just do a brief overview of SMA genetics, because it really helps to understand how these drugs work and how they could potentially work in combination. And so here you're seeing the SMA genetic region, SMN1 and SMN2. So if you go to the next slide, Sarah. And so in the case of SMA, there is either homozygous loss, so both copies, the copy from each parent is lost, or there's some kind of mutation rendering SMN1 unfunctional, and so people with SMA are forced to rely on SMN2. And as many of you are aware, SMN2 copy number influences disease severity, and that the more copies of SMN2 an individual has, generally the less severe disease or the later their symptoms start with SMA. And so that's because there's a single nucleotide change. So if you think of a gene like pieces of a puzzle that need to be put together, that nucleotide change causes one of the key pieces of the puzzle to be missing. So the majority of the protein, of the SMN protein produced from SMN2, isn't fully functional. And that's represented by the pink in this diagram. It's not bad, it just can't replace the loss of the protein that's produced from SMN1 predominantly. So next slide. And so the current FDA-approved therapeutic strategies or drugs are what we call SMN dependent, and that they function by trying to increase levels of fully functional SMN protein to modify disease. And those are gene therapy, so replacing the missing SMN1 gene via a viral vector like Zolgensma does. And then there's splicing modulation, so trying to coax that SMN2 gene to make more of those fully functional SMN protein that contains exon 7, so that missing puzzle piece. And those are Spinraza and Evrysdi, both try to increase SMN protein production from SMN2. Next slide. But when we think about novel therapeutic strategies, we think about SMN independent strategies. So things are like neuroprotection, protecting the nerve itself from injury or from degradation. We think about neural transmission. Can we get that nerve to talk to the muscle better? We think about regeneration. Can we promote that nerve to regenerate and grow and re-innervate muscle to improve function? And when we think about the muscle side, we think about enhancing muscle to restore, help prevent loss of motor function. We also think about things like genetic modifiers, what other disease or what other genes may be impacting disease severity. And then we think about combining all of these different approaches with SMN enhancing approaches to achieve greater therapeutic benefit. Next slide. And so we know that it will likely take a combination of these therapeutic approaches to achieve the greatest therapeutic effect. And when we say combination therapy, we're really defining that as 2 or more therapeutic agents, usually drugs or biologics, that work through the same or different mechanisms of action that are used concurrently. So you can think about an SMN upregulating therapy with a muscle drug or something that offers some neuronal protection. Next slide. And so it's very exciting to see that we currently have 6 drugs in clinical trials, and you're going to hear from NMD Pharma shortly about their muscle drug in trials. So I just wanted to highlight all the exciting clinical trials that are happening. If you go to the next slide. And so briefly, before I turn it over to NMD Pharma to introduce you to them, I want to share a few resources that Cure SMA has. The first being a new clinical trial finder widget, which allows you to really sift through all the various clinical trials. You can filter by things like SMA type, where you're located, how far you're willing to travel, your treatment status, and other things that may fit you or your child or your loved one's preferences. And it can help you whittle down the clinical trials out there to those that you may be interested in participating in and that would fit you for their eligibility criteria. Next slide. And then these are a few other resources we have as well, some from FDA and some that you can find on the Cure SMA website. And we'll make all of these links available later. But without further ado, I'd like to turn it over to NMD Pharma to share more about their company and how they're working to meet some of the unmet needs in SMA. And I'll turn it over to you, Thomas.
Many thanks, Jackie, and a big thanks to the entire Cure SMA team for hosting this event. We are very excited to do this presentation. We have been looking forward to it. Also, a big thanks to all of you who joined this webinar and for sending us excellent questions in advance. We will try to address them all during the presentation and towards the end. My name is Thomas Grønnebæk. I work as a clinical scientist for NMD Pharma, and I will be taking you through this presentation together with my colleague Vera Kiasova, our medical director. I will kick off the presentation with an introduction to NMD Pharma and present to you our history, pipeline, the mechanism of action, and some very exciting data that we recently published in Science Translational Medicine. Then I'll be handing over to Vera, who will present a phase 2 study with our lead candidate NMD 670 that we are currently conducting in ambulatory adults with type 3 SMA. Next slide, please. So NMD Pharma is a Danish biotech company. It is located in Aarhus, Denmark, which is just beneath the Aarhus University logo on the map to the right. NMD Pharma was founded in 2015 by our CEO, Thomas Holm Pedersen, who is pictured in the upper right corner of the picture on the slide. Thomas Holm Pedersen was at that stage, and actually still is, an associate professor at the Muscle Laboratory at Aarhus University. And this is a Nobel Prize winning laboratory where the focus of the research for more than 50 years have been on muscle activation and regulation. It was also at this laboratory that Thomas discovered the importance of the CLC1 chloride channel for transmission of signals from the nerve to the muscle. And it was based on these discoveries that they formed the hypothesis on which the company was founded, namely whether inhibition of the CLC1 channel can be leveraged therapeutically to improve muscle function in neuromuscular diseases where the ability of the muscle to respond to signals from the nerves are impaired. Next slide, please. So today, NMD Pharma is a clinical stage biotech company. Our headquarters is still situated in Aarhus, right across the street from one of Northern Europe's largest university hospitals. Last year, the company raised a Series B of approximately $80 million from a strong investor syndicate that have allowed us to expand our clinical development program and search for new targets and treatments. At NMD Pharma, we develop unique and complementary small molecule inhibitors of skeletal muscle-specific CLC1 chloride channels. And this rapidly restores muscle function by enhancing the neuromuscular junction transmission. We use neurophysiological biomarkers to identify new diseases that may be potentially relevant candidates for CLC1 inhibitors. And we have recently demonstrated clinically meaningful and statistically significant effects with a CLC1 inhibitor in patients with myasthenia gravis. I'll talk to these results in a minute. Please, to the next slide, please. So this is the NMD Pharma team as of today. The picture was taken just a few weeks ago when the entire company gathered in Denmark for an offsite. And we were so lucky to be joined by Jackie Glasscock from Cure SMA and Alison Moore from the HNF Cure, an advocacy group for hereditary neuropathies. And we were able to learn more about the unmet need of people with SMA and CMT, and also how we can collaborate with these organizations to get new drugs available to people with neuromuscular diseases. Next slide, please. During this offsite, we were also able to take a moment to focus on our company values, which we defined in 2022. And these are values that guide the company on our mission to be a world-leading biotech company in neuromuscular disease. I specifically want to highlight the overarching company value, which is that we are passionate about movement. And we use this as a guide on our vision to contribute to a world where people with neuromuscular diseases can live more independent lives. Next slide, please. Our pipeline today consists of 2 clinical CLC1 inhibitor candidates. Our lead candidate is NMD 670, which is in phase 2 for the treatment of myasthenia gravis and spinal muscular atrophy, and very soon will be in phase 2 for the treatment of Charcot-Marie-Tooth disease. The company has another CLC1 inhibitor candidate in clinical development, and that compound is called 1343, and that is currently being studied in a first-in-human phase 1 study. And finally, we have a preclinical candidate for an undisclosed neuromuscular disease. Next slide, please. Now let's zoom in on our target and the mechanism of action of NMD 670. I already told you that NMD 670 is an inhibitor of the CLC1 chloride channel, but what I've not shared yet is how we can leverage this therapeutically. CLC1 is a chloride channel that is specific to the skeletal muscle, where it is located in the muscle membrane. The specificity is great for drug development, as this greatly confines the effect to the target organ, which in this case is the skeletal muscle. The physiological role of CLC1 is to inhibit transmission of signals from the nerve to the muscle. And during intense exercise, the channel is inhibited, allowing sustained muscle activation. The CLC1 is a novel and genetically validated drug target. In fact, in patients with loss of function mutations in the CLC1, you see myotonia, which is difficulty relaxing the muscle after contraction, or essentially excessive muscle contraction. And this is exactly what we can leverage when inhibiting the CLC1. Next slide, please. So there are a variety of neuromuscular diseases that are characterized by impaired transmission of signals from the nerve to the muscle, and where you see impaired muscle function as a result of that. They can broadly be categorized into diseases where the transmission dysfunction mainly stems from impaired ability of the muscle to respond and receive signals from the nerve, and these are referred to as postsynaptic disorders, and diseases where transmission dysfunction mainly stems from a weakened nerve signal, also referred to as presynaptic dysfunction. SMA would historically mainly belong to the presynaptic category, but more and more evidence actually points towards that also postsynaptic deficiencies plays a role in SMA. Regardless of whether the impaired communication between the nerve and the muscle is due to dysfunction in the nerve or the muscle side, inhibition of the CLC1 can increase the muscle's ability to respond to the nerve signals. Next slide, please. I want to just spend a minute to tell you about the results that we have already gathered with NMD 670. NMD 670 has been tested extensively in preclinical pharmacology studies, confirming the ability of NMD 670 to enhance transmission and improve muscle function in different animal models of neuromuscular disease, including SMA. NMD 670 has also been taken through a phase 1 first-in-human study in healthy volunteers, where we showed that NMD 670 was safe and well tolerated. In parallel to the phase 1 study in healthy volunteers, we also did a proof of concept, proof of mechanism study in 12 patients with myasthenia gravis. And that study we just published results from last week or the week before, and you have the reference in the upper right corner here in case you want to have a further look. The study essentially was a 3-way crossover study where we gave 12 patients with myasthenia gravis a single dose of NMD 670 at 400 milligram or 1200 milligram or placebo. And after a washout period, the patients received all the 3 treatments. We assessed safety and tolerability, and we used the quantitative myasthenia gravis score, which is a tool that can ascertain muscle function across muscle groups, somewhat similar to the revised Hammersmith, which is more frequently used in SMA. Next slide, please. And these are, next slide, please. These are the main results from the study, where we showed that patients on placebo, which on the graph here is visualized at the red line, had worsening symptoms during the day, whereas patients that received NMD 670, either 400 milligram or 1200 milligram, improved during the day. And the symptoms that we looked at was symptoms of grip weakness, dysarthria, which is difficulty speaking, ptosis, which is drooping eyelids, and double vision, all showed improvements on NMD 670. And this, for the first time, confirmed the mechanism of CLC1 inhibition in humans, and is obviously very, very exciting and warrants further studies with this compound, both in myasthenia gravis, but also other diseases that are characterized by neuromuscular transmission dysfunction, such as SMA. Next slide, please, and I will here hand it over to Vera to continue the presentation.
Hello, and thank you. To hear me well, my name is Vera Kiasova, and as Thomas kindly introduced me, we are colleagues at NMD Pharma, and I'm the clinical development director for this program. Next slide, please. After this exciting results on myasthenia gravis, and probably another click, Sarah, please. So I'll have the entire slide. Thank you. So now closer to the topic on why you are gathered here today and how our compound can be interesting and can potentially help patients with SMA. And the rationale for the study we are currently performing is the following. We do know that the neuromuscular junctions are defective in quite a significant portion of patients with SMA. There's also been a very interesting publications and continuously explored data showing that patients receiving nusinersen do continue showing deficits in clinical measures correlated with measure of neuromuscular junction function. So based on this and also on quite robust preclinical data coming in-house from NMD Pharma, combining all this together, we do believe that enhancing the neuromuscular junction function in patients with SMA may improve performance, may potentially help to reduce physical fatigue. And we do know, and now you understood from Thomas' explanation, that NMD 670 enhances neuromuscular transmission. So when we designed this study, when we've been working on putting this protocol together, the thought was that SMA patients, all those of you who are connected today and those whom you know, even having the therapy, might have potential benefits from an add-on symptomatic therapy that will target specifically neuromuscular junction. So the Synapse SMA study, the one I'm going to talk to you about, will evaluate the efficacy of muscle strength and function, safety and tolerability of NMD 670. Next slide, please. So this is a phase 2 trial, and I'm sure with the community is yours, there's probably no need even to explain the phases of the clinical development. But where we are now, we're really in a quite short duration phase 2 trial in ambulatory adults with SMA type 3. Towards the end of this month, we have already 28 sites open and active in North America and Western Europe. So what you can see presented here on this map, we have 2 sites in Canada, 11 sites in US. And on the next slide, please, you'll see even better probably visually. The letters are small, but you can more or less situate, how the sites are distributed. And I'm also sure that in the end of March, our colleagues from Cure SMA have also shared with the entire community the exact list of the sites and on the contact person to whom you can address if you are interested in participating in this trial. And we will certainly share after the webinar, together with all the links Jackie was talking about, the contact details for you if you are interested to go to the site, to talk to the PI, to have more information about the site. Next slide, please. So now a bit more on what the study looks like, how long does it last, what we're evaluating. It's a phase 2 multi-center randomized double-blind placebo-controlled 2-way crossover study. And this 2-way crossover study might probably sound a little bit different from some other designs you have seen previously, but that's an interesting and quite attractive explanation to what it means. So in this study, as you can see on the scheme, we start by almost a month of a screening period, then treatment period 1 that will last 3 weeks, in which the patients who are enrolled either receive placebo twice daily or the active compound at the dose of 400 milligrams. After the end of this period, there is a washout period, so no medication is received, neither placebo nor active. And then the crossover, so which means whoever participates in this trial, whoever is enrolled in this trial, will either in the first or in the second part of the study receive an active compound, again for the duration of 21 days, and then 7 days follow-up. So the study is enrolling, it's not only will enroll, is already actively enrolling patients with SMA type 3 ambulatory who have neuromuscular junction deficits aged 18 to 75. Next slide, please. The key facts that I think are important to all of you who are interested is that the study is not so long. It's really, the duration is 8 to 13 weeks. So basically, depending a little bit on the length of the screening period, there are 6 on-site visits. And the important thing is that we are not asking to stop your current treatment. What we are asking our patients are to be on the stable dose, and your physician or the study coordinator whom you'll contact, they'll clearly explain what the stable period means. But the key fact for you is that no need to stop the treatment. You will also take these medications, or also it's twice daily tablets. And as I mentioned earlier, due to the crossover design, all participants will at one point, either in first or in the second period, receive the active drug. Next slide, please. So to make it a little bit more visual, or who knows, probably it's even more complicated this way, but the visits, the way they're presented here is there are 2 visits on the site. You see visit 1 and 2 are on the site. That will be followed by the phone calls, then another visit on the site, washout period, and similar plan of site visit, 2 phone calls, and 2 other site visits. So during the study, you continue receiving your usual SMA treatment. Next slide, please. And now a little bit more into details, because that's exactly not what is always in detail described problem clinical trials where you have already looked at. And just for you to situate a little bit, whether it's for you, whether it's feasible or not. So the age of participants is 18 to 75. Type of participants and disease characteristics, type 3 SMA. We are looking for ambulatory patients who are able to walk at least 50 meters without walking aids. And this is something that we are looking at during screening period while performing the 6-minute walk test. We are also, as you understood from Thomas's explanation, and as Thomas and also Dr. Crawford will explain a little bit later, due to the mechanism of action as well, we are looking for the participants with partial neuromuscular junction transmission defects. So this is not a usual examination for that you are used to as patients with SMA, but this is something that is used at screening for patient selection, and participants with genetic confirmation of diagnosis when needed. We have a certain limitation on the BMI, ranged 19 to 35, and also we are asking for contraceptive barrier requirements consistent with local regulations. Next slide, please. As for the key exclusion criteria, of course, you know very well that in the protocol there will be an entire longer list, but the key are medical, from perspective of medical conditions. We are excluding patients, or it is exclusionary for the participants to have prior surgery or fixed deformity, scoliosis, contractures, which would restrict ability to perform the study-related tasks. It's also exclusionary for the participants with other significant diseases that may interfere with the interpretation of the study data, which means other neuromuscular or muscular diseases, current or chronic history of liver diseases, breast cancer within the past 10 years, or any other malignancy within the past 5 years. Positive drug screen as well is exclusionary, and as well the evidence of clinically significant lens opacity or cataract, or an inability to undergo the ophthalmologic evaluation at screening. Next slide, please. So what are we planning to look at in this study? From the inclusion criteria, you guessed that there is a 6-minute walk test, and the ambulatory patient who need to be able to perform 50 meters at the inclusion when the test is performed. And what we are planning to look at the end of the study as the primary objective is the change in muscle strength and function after the administration of an NMD 670 in the dose of 400 milligram twice daily for 21 days. And this change in muscle strength and function will be compared to the participants compared to participants who received placebo. Next slide, please. And to a little bit, give you an overview of what to expect if you are interested and you decide to participate in this study. Some other procedures that will be performed during this 21 days or the screening period are those like handheld dynamometry that you can see here with the picture representing a little bit how it happens. The revised Hammersmith scale, that's what also Thomas mentioned when he was presenting the QMG for the myasthenia gravis. And because we are interested in the neuromuscular junction deficit, so there's a single fiber EMG as well that's performed at certain sites. Next slide, please. Other study assessment and procedures and what we are interested to look at, of course, one thing are objective measures and what the regulators or what any clinical trial is measuring. But another thing which is particularly important to us and to you as the patient community is also evaluating the quality of life. How administering of this compound in addition to a current treatment might improve certain parameters of your quality of life. How do you feel on the level of the fatigue? That's why we're going to use the fatigue severity scale. And of course, the list described here of other assessments, quite typical for all the clinical trials: height and weight, physical examination, vital signs, ECG, and following all the safety parameters because it is a phase 2 study. So we are certainly looking at efficacy, and we are particularly evaluating also the safety and tolerability. Next slide, please. So the current enrollment status, where are we? You've seen that there's quite a number of sites opened, and we are actively marching towards the recruitment. The recruitment is competitive. It's true that the sites in Europe started a little bit earlier, so the first around 5 to 6 patients were mostly recruited in European sites, and now that almost all the US sites are active and are doing their job very well. We're recruiting patients now, but there is still an opportunity for all of you to jump to contact. The recruitment is competitive. So once we're getting to 54 patients, that's our target right now, the recruitment will be closed. And it's true that the question we are looking for the patients to participate in this trial, but what are you looking for as well? Because most of you are probably already on the treatment and still feel that some other things are needed to be improved. Of course, according to our hypothesis, and it's a hypothesis, more muscle strength can be developed with this compound, less fatigue. Next slide, please. And with the full transparency, and it's not about over-promising, and we are in the research setting in the phase 2. But what we know so far, as Thomas already shared, is that NMD 670 was already studied in patient population. It was shown to be safe and effective, even after 1 dose. The improvements that we observed were in muscle strength. And another attractive, interesting, let's say, fact for you is that it's a short study. It's not one of those long duration, several year studies we often see with larger studies. It's short, so you won't really have to travel that often to the site. And if traveling is needed, then the support can be provided. So this is also something we kind of thought about. Next slide, please. And to give you a hint on what's happening right now, you've seen that there's quite a number of sites already. And now in May and the first week of June, we are opening 2 additional sites in US. So for those, if you can't really see some places close to you, there will be a site in Denton and in Baltimore soon activated, so also giving more opportunities. And next slide, please. So if you are interested in learning more about the compound, more about the company, about the trial, it's new, it's different, so don't hesitate to reach out to study coordinators if you really want to get more details on the trial. You can also follow the updates on this study and on other studies we are starting with the compound on clinicaltrials.gov. Now that you've seen our faces, both Thomas, who's been in the company longer, and me during this webinar, please don't hesitate to write an email, ask a question, and we'll be happy to provide more details. And for sure, as most of you know, in the beginning of June, there will be the biggest event, the Cure SMA annual conference in Austin, where we will be present as a company, as a team, and we'll be certainly happy to answer following questions and just to have a discussion also with community. Next slide, please. As Thomas mentioned, we did receive a lot of questions, but probably before just jumping into the questions, I would like to pass the floor to Dr. Thomas Crawford, who kindly accepted to join us during this webinar to provide his opinion, his view on the compound, on the different things happening, and happy to hear.
So thanks for the invite. A couple of things that I think is probably worth emphasizing. There are two big categories of therapies for SMA. There's the disease-modifying therapies. We've now summarized this as DMT, things that are going to slow the rate of degeneration down. And so those studies take a long time because we don't see changes in SMA day to day. We see it over months or even years. And so the therapies for all the SMN enhancing drugs were DMT, and we had to start something today and then watch and see what happened a year from now and see if the kids that got the medicine were better off than the ones that didn't. And there's obviously no potential for a crossover design. This is not a DMT. This is not something that's going to modify the course of the disease. It is something that's going to work for the duration that you're giving the drug, and so this permits a different kind of much more efficient trial design. This crossover thing where you take the drug or the placebo for a while, wash out, and then you take the placebo or the drug for a little while, and we see what the differences is between those two to see if there's something better. This drug probably does not have a tolerance effect, so that means that we can't promise this yet, but it seems unlikely that there's going to be a tolerance effect in that as long as you're taking the medicine, it would work, and when you don't, it doesn't. Another thing that's probably worth pointing out on this trial is that I think it's likely that although many people would be interested and we'd love to have them volunteer, maybe half of folks won't have the eligibility criteria. And that sounds very disappointing until you think about it. The eligibility criteria are designed to find people in whom we can measure a benefit. Now, measuring a benefit is not the same thing as experiencing a benefit. It means that you're in that category of people that we can say that we can see a difference between now and 3 weeks later in a measurable way. Obviously, you cannot get a drug approved unless you can measure a benefit, and then we have to show that that benefit is meaningful. I think there's every reason to believe that the drug effect will be broader than the measurable effect, but we can't get the drug to everybody until we are able to measure a group. So for those of you who sign in and we are not able to show that the changes in decremental response, that doesn't mean you didn't contribute to the trial. You absolutely did, and we'll have to see how it is we can get it to to the broader group at the level of labeling or licensing. So there's that. And then I want to point out that probably the decremental response is seen in people who have expanded motor unit sizes. When a motor neuron dies, the muscle fibers next to it or that were previously innervated by it are orphaned. They send out those muscle fibers send out a distress signal. Help, I've been orphaned, I'm lost. Send me a line, and the neighboring motor neurons will send out a twig, a sprout to rescue it and bring it back in. Those lines that are attached to the extra ones that were rescued are probably a little bit more feeble, and those are probably the ones that will boost most with this therapy. So I think that there's reason to believe that as the DMT go further along and we have more and more kids, especially the ones with single copy, have large motor units, I think there's every reason to believe that this drug will be beneficial at a much broader range than the original criteria, the sweet spot where we think it's possible to show the benefit. So those were the specific bits that I wanted to add to the presentation before. Why don't we broaden it up? Am I alone on this? There we go.
No, we're together again. Thanks a lot. And I think it introduces very nicely also the answers that we would like to provide today to the questions, because we realized also that a lot of questions that came from the participants of the webinar today were about expanding to a larger population, going large. And thank you, you really put it nicely. We need first, the stage where we are, we need to show that it works. To show that it works, we need to go in a smaller targeted population. But just to, the questions are here on the screen, but whether this can also help people with SMA type 3 who are no longer ambulatory, based on what we know and what we do believe, this can. It just has to be there are step-by-step exploration, and we are in the step where we need to demonstrate at a smaller population that it works. We go back to the questions that we received and that we wanted to address, because there were a number of them, so it's a little bit of a shortened list, but there were several people asking on whether we are having plans to extend the trials to children. And indeed, the answer is, in the same direction. There are regulations. We're currently in the phase 2 in adults, where we need to demonstrate its safety and efficacy before starting afterwards expanding our trials to kids or to younger kids, to adolescents. And this also probably can, from this I can jump to question number 4, because there were several people asking as well whether there will be follow-up trials where the participants have been dosed with Zolgensma. And actually, if you do look at our protocol and you decide to go there, you can see that this is one of the treatments that we do list as an authorized one. So because if it is authorized by the kids for the kids, so even if it happens, it's going to be a little bit later. But yes, the answer would be, if there are follow-up trials, we will anyway always have the list of the 3 authorized treatments. And
one thing, I think for those first 4 questions, I think it's important to realize that the use of the DMT is probably conditioning the motor unit system to be more successfully treated with this therapy. And so I think I'm very bullish on the idea that now in the new era of DMT treated SMA, this is the next step to be able to make those partially recovered motor neurons work even better.
I think certainly we're on the same wavelength on this with you in the way we are looking at the compound. And the last one said from me, and then I changed to our NMD Thomas. There were some questions on, more in the inclusion-exclusion criteria and why we're having a certain BMI limitation in the clinical trials. and whether it would be in the label if the drug is approved by FDA. And getting back to the beginning of discussion, we are currently limiting to the BMI just within this trial, and actually we're not even limiting, we're expanding, because since the study started, we tried to open up a little bit further, trying to give the opportunity to more patients to enter. But this has nothing to do from now on until when hopefully successfully it is approved by the FDA. So Thomas, to you.
Sure, thanks, Vera. So we've had two excellent questions around the ability of the compound to build back muscle mass, and also whether we can expect to lose the strength that we gained with the medicine. I'll tackle the first one first. So we don't know at this stage if the compound can have any muscle building effects. We know as of today that in preclinical models of neuromuscular disease and also in patients with myasthenia gravis, we can increase the strength of the muscle. We have a hypothesis that when you increase the strength, you also increase the ability of the person to be more physically active. And that could have a muscle building effect when given over the longer term. So this is certainly something that we will be studying in our trials, but the answer as of today is we don't know. Would we expect to lose the strength gained if the medicine stopped? And that question actually ties nicely to the previous question, because the answer is the mechanism of action of the CLC1 inhibitors is acute, and we see acutely an increase in muscle strength that will disappear if you stop the treatment. But if there is a conditioning effect, if there is sustained effects, and whether that is related to an increase in muscle mass when given longer term, or whether it's due to other mechanisms, could be re-innovation, it may be that some of the strength is maintained even if you stop the treatment. But these are questions that we continue to explore both in preclinical studies and in the clinic. Vera, maybe back to you again for the final question.
Sure, if Tom has nothing else to add.
No, there's a standard array of side effects that that you have decided to look for. This is pretty much a boilerplate of reviewing various organ systems to try to figure out what problems there might arise. I might point out that on the mechanism of action, we're trying to inhibit a CLC channel that occasionally people are born without. And the people who don't have that, who are born with a genetic disease called amyotonia congenita, who don't have that channel at all, live a long life. They have neuromuscular problems. They build up too much muscle, and they actually look rather healthy in appearance, but they can't relax effectively. And so we don't have to worry about this drug being overdosed, at least not on the effect size of it, because that's not a problem that we haven't experienced in spinal muscular atrophy. So there's the good news, that the side of that. Now, whenever we're looking for side effects, we're looking not only for whether or not the effect is there, but we're looking about whether there's off-target effects, and those are the things we have this standard array of things you're looking for to see if the problems that arise as a consequence. But there's again reason to believe that the problem that those people who are missing the channel have is kind of exactly matched to the defect that the people with SMA who have these feeble sprouts don't have. So it is kind of a hand in glove a way to think about making large motor units work better.
Thank you, and very well spotted. And it's true that, as you say, we do look at all the system organ classes, and based on the results that we have from the phase 1 studies, we can at least that now we can certainly say that we compared the safety profile, and we know that there was nothing more important with the compound than without compound. So what we are monitoring indeed is the myotonia that you mentioned, and there are possibilities also for the patients to monitor and to report things on them. And then all the standard safety assessments that are done in all the clinical trials. But we do believe so far that there is no potential, I mean, there's still ongoing preclinical studies as in every clinical development program. But at the level of knowledge that we have right now, we certainly have a certainty to believe that no particular adverse events would happen at the doses that we are currently using for this trial. So I think we are getting to the end of our lunch hour and webinar. And from what we understood is the first time webinarists with Cure SMA, there will probably be further questions that would arrive during the webinar to the organizers in the chat that we actually don't see, but we'll certainly receive them after the webinar. So it's also more message to the public. Don't feel lost or unanswered if any of the questions that you've sent earlier was not taken today, or you haven't got your answer during the presentation. We'll do our work, our homework after the webinar, and we'll certainly share with you once again all the information about the study, all the sites who are recruiting, or as Dr. Crawford's John Hopkins, soon to join the study, will be recruiting.
Yeah, we're on soon.
So you see now, you will be in the same boat. So thank you very much.
Are both of you two?
If Sarah, if I skip for the next slide, it will be a thank you slide, but it's true.
Are both of you two coming to Austin?
Yes.
Yes. Are you two coming to Austin?
I'm coming to Austin. And
you're joining Austin. I will not be there, but we'll have other team members
Okay.
in Austin as well.
Well, we will look forward to giving you trouble there.
Yeah, will have to be another time.
So let us finish on the positive note, showing that things do advance, and we were really happy and pleased to have this time today, to have so many participants sign and see the interest from the community. And please don't hesitate to reach out to either by Cure SMA, or now you'll have our emails, and we'll be happy to follow up and share